Okay, we can go ahead and get started. Hi, my name is Melanie Rubin, and I'm the Director of Senior and Adult Programs at the Lawrence Family Jewish Community Center, Jacobs Family Campus. Please note that we are recording this program. We're happy to be collaborating with the Sanford Burnham Prebys Medical Discovery Institute on this three-part series of the science behind women's health. And many thanks to Bobby Laraga for helping to arrange the speakers. Today, we will focus on heart health with Karen Okur, PhD, Assistant Professor, Development, Aging and Regeneration Program, Neuroscience and Aging Research Center. Dr. Okur's lab is working to understand the cellular and molecular basis of heart disease, and one project is focused on the genetic basis of atrial fibrillation. She will take questions at the end, so please put them in the chat feature, and we'll try to address as many as time permits, or we'll send them to her in a follow-up email. Next Wednesday, June 16th, the topic will be breast health with Svasti Harikaran, PhD, Assistant Professor, Aging, Cancer and Immuno-Oncology Program, the NCI Designated Cancer Center. I'll put the registration link in the chat so you can register for that one as well. I'm going to turn the spotlight over to you now and check the chat for further information. Thank you.
Hello, everyone. Thank you for attending, and I'm very excited to give you a little bit of a glimpse into the kind of research that my lab does. If you had a chance to read the cartoon already, you've already understood a couple things about how we look at heart disease and how we try to understand it. And that is, we use lots of different model systems like fruit flies, which I'll be telling you about, but also mice, frogs, monkeys, people, even fish. And the other thing that cartoon brings out is the lack of focus on women in a lot of these studies, not just women humans, but also female monkeys, mice, etc., because of the addition of hormones that complicates the ability to evaluate the data. But in any event, I'm pleased to tell you that in our lab, we primarily look at females, and you'll see why in a few slides. So my lab studies heart disease, but before I get into that, I want to give you some details on human heart health. And this I guess in general, when you talk about heart disease, everyone conjures up a man keeling over from a heart attack, but heart disease is also the #1 killer of women. And it is #1 ahead of cancer for both men and women. It's consistently the leading cause of death in industrialized countries, including the US. And in the US, 606, almost 700,000 Americans die every year from heart disease. Now, that means one in every four deaths are from heart disease, and they occur about 1 in every 40 seconds. About 66% of cardiovascular deaths occur in people age 75 and older. So it's an aging-related disease, and that means when you do your studies, you have to take age into account as much as possible. But a very disconcerting set of statistics has come out of some recent studies, and in particular, over the last 20 years or so, myocardial infarctions have steadily increased in young people. So this was a cohort of 30 people, 35 to 54 years old. And this is very disconcerting and may be related to the increase of incidence of obesity. Overall, the myocardial infarction related emissions went up from 27 to 32% of all admissions in this age group. And interestingly, women admitted for a myocardial infarction into a hospital increased from 21% of all the MI admissions to 30%. That's an enormous increase. Now, men also increased in terms of the percentage of admissions in this age group from 30 to 33%. But women, as you can see from this statistic, are catching up. So this is something that obviously we need to pay attention to. It's been speculated it may be related to the increased stress, as women are more likely now to be both balancing work and family-related duties, and that the increased overall stress levels in their lives may be contributing to this disproportionate increase in women with heart problems. So what are the risk factors? Well, they're all the ones you've heard about probably over your entire life and all your interactions with your doctors: high blood pressure, high cholesterol, smoking, diabetes, and family history, which means your genes. And that's what I'll primarily be talking about today. But my lab, oh yeah, there's a couple things that we can do about this. I can't tell you anything about your medical responses, but obviously exercise is a real big no-brainer to try and deal with high blood pressure. But it turns out some recent studies have shown that if you get more sleep, you can reduce your blood pressure. And so both of those are real easy things to recommend to do to deal with that particular risk factor. And of course, just say no to smoking. So those two are big, big things that anyone can do and recommend for mitigating your risk of heart disease. In my lab, we look at some of these other aspects, these other risk factors. We look at the effects of high-fat diet and high cholesterol on heart function. We also look at the pathways, genetic pathways that contribute to diabetes. Those are known, and we try to relate those pathways to the heart disease we document in our fruit flies. What I'm going to talk about today is how we are looking at how we use this genetic model system to understand the underlying genetics of arrhythmias in the heart. So that will be sort of the focus of my talk. However, before I get there, I just want to put in one more bit on women's heart health, because this is an important one. There's a lot of common heart attack warning signs. Of course, chest pain is the most common symptom overall, but there's a bunch that are more common in women than in men. And these include things like being lightheaded and suddenly nauseous, back and jaw pain, which isn't usually typically associated in most people's minds with a heart attack. Arm and shoulder pain, maybe a little bit more, but then finally shortness of breath, as if you just suddenly finished running a marathon. All of those are more common in women than in men. So you women out there, pay more attention to those aspects if they should suddenly occur. So what causes heart disease? That's the bottom line, the thing my lab is trying to understand. A lot of our initial understanding of heart disease came from an early study called the Framingham Heart Study, which was conducted on about 5,000 men and women who lived in the Framingham, Massachusetts area. And it was started in 1948 and continued over about 40 years. And they just checked on everyone every 2 years. They had medical exams and psychological exams and write-ups and questionnaires. And they assembled all of this information. And from this came some of our very early understandings of how heart disease occurs and what are the risk factors. In fact, those identified risk factors that I mentioned earlier came out of this study. But what also came out of this study was our understanding of heart disease increasing with age. It was an incredibly clear trend and undeniable. But what was also interesting that came out of the study, because it's one of the few that actually are centered on women or have women as part of the cohort, was that women had a lower incidence of heart disease than men over most of their lifespan until they hit menopause. And then they caught up and began to exhibit the same level of heart disease as men. So this gave us some of our first early clues that hormones and just general hormonal signaling in the body could play a role in heart disease. So why use a fruit fly? There are plenty of people who have asked this question, some of them famous. But we can use the fruit fly to get answers for a number of reasons. One, because they have a heart, and that's a pretty important thing if you're going to study heart disease, and I'm going to show you an example of a fruit fly heart shortly. But more importantly, fruit flies have 75% of the genes that cause disease in you. So if there's a particular disease we want to study, almost certainly a good chunk of the genes that underlie that disease we can test in the fruit fly. Another good thing about them is they're small, they're cheap to keep. Mice cost a lot of money, and hundreds of thousands of dollars over a 4-year grant, whereas a few thousand a year for fruit flies. So they become a very tractable and affordable model system for researchers. Flies age rapidly. This is perhaps one of the real big advantages. So if you want to look at aging in mice, it takes up to 2 years to get a mouse to be old. Whereas flies, 1 week is about 10 years of a human lifespan. And so within a couple months, we have the equivalent of an 80-year-old fly. And the most important thing from the perspective of our research is they have loads of genetic tools. And so here's the heart model. This is our, what we call our denervated fruit fly prep. So what you can see here is the abdominally located heart in red in this schematic. And then down here is a fruit fly that has been basically denervated. We cut all of its head and its central nervous system off. You can see the wings sticking out a little bit, and then the striped part is the abdomen. We've taken all the guts out. We've taken the ovaries out because this is a female. And what you're looking at is the beating heart tube. And it doesn't look like a human heart, but it has 4 chambers. And in fact, your heart looks like this during the very early stages of embryological development. And importantly, many of the really crucial genes and proteins that make hearts function, we find in the fruit fly. So what we do is we focus in on this area in the red box, and we take high-speed movies of these beating heart tubes. And what you're seeing here is a subsection of the heart, but underneath is something called an M mode. That's what your doctor will do with, if he does an echocardiogram on you, he will take an electronic slice through this movie and then do that for every frame in the movie, line them up, and what he can do, or she can do, is see the movement of the heart walls. And you can see that here. This is the heart wall contracting in and out as the heart beats. So this entire M mode is the entire movie that you're watching. And you can see that this is a really regularly beating heart. It's a young fly. It almost always, their hearts are quite strong and rhythmically beating. Now, why do we use females in our studies? Well, this picture should be able to give you a pretty good clue. This is a female on the left, and this is a male on the right. And you can see the females in flies are much bigger than males. And when you're dissecting hundreds of these fruit flies in a day to do your experiment, these are a lot easier to dissect apart. But we've recently come up with a method to look at these, you'll see in a minute, to look at the heart in both flies without dissection. So now we can go back to looking at both males and females. But if we want to look at a particular gene, what we can do is we can alter the gene function specifically in the heart. And here what you're looking at is a heart that has been mutated for a gene that's been associated with atrial fibrillation, strongly associated. And this gene is called KCNQ. It's a gene that makes an ion channel. And ion channels are what allow the electrical activity of your heart to be conducted. And what should be pretty obvious from this movie is that the heart is fibrillating, and you can see that in the M mode underneath. You can see these rapid contractions of the heart. Now, if this happened in a human or in a mouse, they would immediately have heart failure, and unless they were resuscitated, they would die. And this is in fact what happens when you've probably all heard stories about a fit athlete running down a basketball court and collapsing with sudden cardiac arrest. Almost always, that is a mutation in this KCNQ gene that we've mutated in the fly heart. And you're seeing what happens, what that fibrillatory event is, but over and over in the fly. It doesn't kill the fly. They get their oxygen from another system. So it makes it really a good system to look at badly functioning hearts, because they don't die. At least not right away. They take a few days. And so, as I said, this gene is really, really strongly linked to atrial fibrillation. And this is just to show you what atrial fibrillation is. It's a change in the electrical activity in your heart. So a normal heart, atria contract, and then the ventricles due to this progression of electric activity throughout the heart. But in atrial fibrillation, the electrical activity has what we call re-entry circuits, and it doesn't pump effectively. And basically, that is why you end up with stroke, because the blood pools and clots, and then sometimes those clots dislodge and cause stroke. But if untreated, it leads to heart failure as well as other cardiomyopathies. So we're very interested in studying this because there is no cure for AF. There's some palliative treatments, but many of those also cause arrhythmia. And basically the only way to cure it is to go in and do an electrocoagulation of the badly functioning tissue. And almost always it recurs. So this is a really big need in the medical community to come up with a way to treat atrial fibrillation. So here's our strategy. Our strategy is to use this fly heart preparation that I already talked about. This is our new one where we don't have to dissect. We have this fluorescent heart. We can just take a movie of it through the cuticle and then analyze the function. And we have partnered with another lab at our institute, the COLAS lab, and they have a system where they take basically skin cells and turn them into cardiomyocytes. And that's what you're seeing here is a cell culture of beating induced cardiomyocytes. And our strategy is to test all of the genes that have been related or have been shown to be related to atrial fibrillation from patient samples, to test all those genes in both of our systems. Our rationale is that what is coming up with common effects in both systems must be a really fundamental way to create arrhythmia. And if we can identify those fundamental mechanisms, we can probably treat them. So the idea is to go through and test all these 200 plus genes, and more are coming through in publications every day, and identify these gene networks that underlie arrhythmia. And if we can identify gene networks, we can design drugs to target those particular networks or subsets of the networks and interfere with the process. And also come up with targets that we can use as predictors of whether you're going to develop atrial fibrillation. So the ultimate idea is to take this huge conglomeration of atrial fibrillation related genes, which is almost impossible to decipher in vertebrates, and figure out from our model systems a more simpler network that we can go in and dissect and come up with mechanisms. And so I have to acknowledge all the people that we work with for these studies. So in my lab, there's Jimmy Kizos is the main driver behind this aspect of our projects. Geo Vogler in Rolf Bodmer's lab, also a collaborator of mine here at the Institute, is the main driver. And Alex Colas' lab, Anais, has also been very strongly involved in this project. And we have a couple other clinical collaborators that we're pulling into this project. But we're very excited that we're going to be able to come up with fundamental networks that are going to make AFib treatable. And thank you very much for your attention. I'm happy to take any questions you might have.
Thank you so much. How fascinating that you can work on fruit flies and it's just a replica of a female's heart.
It shocked me, I have to say. I come from a neurobiology background. When I started trying to develop this system, the commonalities between fruit flies and humans at a fundamental level, at a cellular level, are striking. It's been an eye-opener even for me.
Anybody has any questions, you can either feel free to unmute yourself or turn on your video and wave your hand. You can put it in the chat. I'll start with a question. Are there any natural ways to reverse AFib? Does it ever get resolved without medical intervention?
Yeah, unfortunately, no, at least not to my knowledge. It's really important to get treated because, as I said, this abnormal electrical activity, when left unchecked, tends to become a sort of a snowball effect, a positive reinforcement loop, where it induces other arrhythmic activity, not just in the atria, but also in the ventricles. And it causes remodeling of the heart, structural remodeling. And so it's really important to get it treated as soon as you realize that it is your problem. And hopefully it can be slowed. But if it becomes persistent, chronic, then that's when you have to go in and actually ablate the badly behaving cells in the atria.
Well, there's no way to prevent AFib, huh?
Well, like anything else, you know, all those risk factors that I said, you know, you exercise, you eat and drink in moderation, you get a lot of sleep. All of those things are the way to prevent any heart disease, not just AFib, but they will also help with AFib as well. Yeah, no magic bullets yet, but we're hoping. we're getting there.
Is tachycardia the same as AFib?
AFib is a form of tachyarrhythmia, yes. But it's primarily just in the atria, but it then can induce changes in the ventricles, as I said, as well. But there are different types of tachyarrhythmia. This is just one category.
If both parents had AFib, would I get AFib?
Yeah, that's the genetic risk factor of heart disease. There's always, and we don't know which genes to look for yet. But yes, if your parents have AFib, you have then a genetic hereditary risk factor that means you're potentially more likely to develop it eventually. The good news is that it's not just genes themselves. It's not like a single gene for AFib. It's what we call a multifactorial disease. And we don't have a single genetic driver. What it depends on is the background genetics as well. And so that's why you can have two siblings with the same parents. One develops AFib and the other one doesn't. Depending on all the other genes that they got from their parents, they may interact with the underlying pathway genes to promote or to retard your tendency to get atrial fibrillation. That's that hairball network. So if I go back here, all of these genes have been related to atrial fibrillation. Some of them are probably causal in that they push the disease to become a manifest disease, but other ones may be just sort of giving a little bit of a boost to some of those causal genes. And if you have them, it means you're more likely to get AFib. If you don't have them, you're less likely. And what we want to do is figure out which of these are more causal and which ones are what we call modifiers. And once we figure out that genetic sequence, then we can, that pathway, we can then think of strategies to go in and to either promote heart health or to block the deleterious effects from that particular genetic network. And also, if we can identify the core network, we have tools to go in and become proactive and look at, because now everyone is getting close to having their whole genomic sequenced. Just as a course of your normal medical history, you'll get your genome sequenced. And if we do that, then we can go in and look for some of these marker genes that will let us know early on that someone's predisposed to ultimately get atrial fibrillation. And then you can go in early, and prevention, as the adage goes, is worth a pound of cure. So all of these things we hope will come out of this kind of approach that we're developing here.
Anybody have any questions? You can always put them in the chat or send them privately to me. Will you be sending more of your fruit flies into space in the near future?
Yeah, so that was very exciting. We sent fruit flies a couple times to the International Space Station, and what we found, obviously, we were looking at heart function, and this is where the small size came in really handy.
And then somebody swatted them because they thought they were flies and annoying them.
No, we had to be, they were triple contained, I gotta tell you. There was no way they were breaking out of that jail. But no, we found that microgravity causes cardiac remodeling and changes in the way the heart sort of processes its waste and recycles its proteins. So your muscles, when they beat, proteins get damaged. It's like if you stretch a rubber band over and over again, eventually it wears out. And so what happens is your cells have to kind of break it down, your muscle fibers down a little bit, break them down into the building blocks, and then put them back into a brand new functional fiber and put it back into the contractile apparatus. And we found that process breaks down in microgravity. And we have no idea why, but we have a lot of pathways that we think are involved. And so we just submitted another proposal to NASA a month ago, and we're hoping that we'll be able to get funded so we can do another send another set of flies up to the ISS.
Fascinating, fascinating. Well, thank you so much for your time today. And if anybody has any further follow-up questions, you can always email me. My email was in your Zoom reminder link, and I can forward your questions on to Dr. Okar. Please sign up for next week, the breast health lecture, and then forgot what we're talking about. Immune health will be the following Wednesday. So you can sign up for both of those at the link that I put in the chat. You can learn more about Dr. Okar's research and her bio. I put a chat in the link, and also find more about what the JCC has to offer, including we have some medical fitness programs. So for people who are concerned about heart health and want to have preventative measures, we have all kinds of programs here. And I'd be happy to take some questions about the JCC if you email me or give us a call. So thank you very much for your time today. And if there's no other questions, we will bid adieu.
Thank you, everyone. Bye.
Thank you so much. Bye-bye.